Hello, everyone, and welcome back to our monthly virtual series. You know, I'm Sandra Franco, and I am the Education and Outreach Coordinator for the Center for Integrated Cellular Analysis here in New York City. In this virtual series, we aim to highlight research and methods that stem from our center and that we think that might be of interest for the broader biology community. As usual, at the end of the talk, there's going to be time for you to ask questions, and you can do so by either raising your hand or typing your question in the chat box. With that, I'll introduce one of our speakers today, Farhia Kabeer, who is going to present a joint work from the Shah and Aparicio's lab. Farhia is a postdoctoral researcher at the Aparicio lab. She's a medical doctor who came to Canada to start cancer research after practicing as a clinician in Pakistan. She has recently obtained her PhD from the University of British Columbia in Vancouver. Farhia has a special interest in cancer evolution and the dynamics of genomically defined clones. Her research focuses on alteration in fitness landscape reflected in time-series patient-derived xenografts with or without chemotherapies. She's also interested in gene expression and mapping fitness due to transcriptome variation. With that, I'll turn it over to you, Farhia.
Thank you very much, Sandra, for the introduction. Okay. And thanks everyone for attending this session. Let me share my screen. Okay. So the main purpose of this project was to develop an understanding of clonal cancer dynamics defined by copy number aberrations in the context of therapy as a route to understanding mechanism of drug resistance. This work is published in Nature a couple of months ago, and it was a collaboration between the groups of Dr. Aparicio at BC Cancer Vancouver and Dr. Shah at MSK. I was leading all the biologic experiment and biological team, and Dr. Sohrab Salehi. He developed and applied new models on the data, and he is with us today. Hi, Sohrab. So today I will give some highlights from this paper. And after introduction and experimental design, I will request Dr. Salehi to brief on the new models, and then I will show you the results from one of the main triple negative PDXs in the interest of time. Otherwise, we had like around more than 3 PDXs on which we worked on with 3 main time series and drug exposures. So one of the insights about cancers, we know that they are heterogeneous, and tumor cells, they have property to go on replicating, resulting in fluctuations in the relative proportions of cells. If there is no selective advantage, it will lead to neutrality. But if selection applies, for example, in the form of drugs, the sensitive population will decrease the tumor burden, and the resistant population will emerge, which becomes a clinical problem that could cause tumor recurrence or metastasis and ultimately treatment failure. So we need a conceptual approach first to identify the subpopulations in the growing tumor that could be used to consider relationship between cells based on mutation and cell division as heritable marks that lead to clones. And another way of is to make use of transcriptomic state changes with defined cell states, but we are not going to discuss that today. So in our work, we focus primarily on identifying clonal structure, where a clone is defined as a group of cells that are related to each other by descent through a unitary origin. We can represent the relationship between cells, hierarchy, and infer the order of evolution that's known as phylogenetics. And we made use of these phylogenetics throughout our measurements, and the tendency of clones to increase or decrease over time that leads to an idea that fitness of a clone may be measured by its reproducible growth trajectory, while in contrast, selection is an ongoing system that could be transient or it could be stable. In population genetics, the concept of stochastic drift was first applied on allele frequency where meiosis was taking place, but in cancers, there is no meiosis. But the principle of stochastic transmission of genotypes still applies, and the Wright-Fisher model is one way of defining how to estimate it. Dr. Sohrab Salehi will explain this model in a bit. In this model, we assume that the cancer clone that has the higher fitness will grow to occupy a higher clonal fraction faster than the one with the lower fitness. Then we need to have repeated observations of the trajectories of these clones over time. That links genotypes to fitness. And the reason why it is pertinent to our work is because a landscape genomic study in triple negative breast cancers in 2012, alongside of landscape of all breast cancers, showed that copy number aberrations are very important drivers of the genome, as shown in these panels in representations taken from those papers. But they were done in bulk sequencing and are less well studied as mediators of drug resistance. And the idea of decoding clonal structure was also motivated by the recent development in the paper published from our group and in collaboration with Dr. Shah's group in scalable high throughput technologies of acquiring single cell genomes and defining their copy number profiles. So we set out to map this by taking population genetics approach to studying copy number associated single cell dynamics in time series PDX models and with or without drug pressure. So we asked whether copy number measurements can give evidence of clonal fitness or selection. And if yes, what would be the features of fitness landscape after continuous drug exposure? The time until the time when drug resistance occurred. So we developed models of tumor evolution. And we designed experiments where we took patients' tumors because, like it's not that convenient to have patients serial biopsies from the tumors. Most of the times they got like tumor excision, or there is no residual tumor, or they are at a site where we cannot take the tumor biopsy. So we took patients' tumor and serially and transplanted in next generation of mice and exposed to the platinum, which and I use cisplatin, which is standard cure of chemotherapy and first line of treatment strategy in most of the cancers. So what I did, I took along a tumor after treatment, residual tumor was retransplanted and re-exposed to the drugs in a time series manner till they started showing resistant pattern. So along with the treatment time series, I had absolutely untreated branch in a time series manner as a control. And along with that, I had some samples that at the same time point kept as in treatment off state. So we can compare the continuous drug pressure and the drug holiday samples. And we took a single cell genome at each time point, or we sequenced around more than 1 million single cell genomes from all the PDXs over many years, and passaging and measuring single cell copy number profiles and computing phylogenetic trees to identify genotypic clones and their relative abundance by applying sitka model for creating the phylogenetic trees, and by applying fitClone model to measure this selection coefficient and the relative fitness of the of various clones in our data. So Here I will request Dr. Sohrab Salehi to brief on these two mathematical models, what we applied throughout our data. Up to you, Sohrab.
Excellent. How would you like to do that? I can share screen, or would you like to? Oh, okay, we can do it this way. All right. So here we, with the explanation that Dr. Kabeer gave about the design of our experiments, the first step is to try to construct a phylogenetic tree, and gives us these latent variables, the clones. But the advantage here is that you have this at the level of a single cell. And the first challenge there would be that, well, you have thousands of cells, and these cells are measured over thousands of genomic features. So we clearly need a scalable method to do this. And sitka is such a method. It's a Bayesian method. If you're familiar with Bayesian phylogenetics, you know that are often what's possible is only a handful, let's say in the low hundreds of species or taxa, over maybe hundreds of phylogenetic markers. But with sitka, we have a scalable method, and in the next step I show why this is scalable. Okay, can you go? Yes, thank you. All right, so there are three main parts to sitka. The first one is the sitka transformation. So what we do here is, once you have experimental setup with these evenly with even coverage coming from, say, DLP plus, we get this in this copy number aberration matrix. So rows would be cells and columns would be bins. But this is highly correlated. So if you were to use this as an input to a phylogenetic method, there are all sorts of problems that you'd face. But what you can do is to transform that into a binary matrix, into this pattern of presence and absence of copy number changes. So this is what's shown in the middle. And just to have it a little bit more clear, and at the very end, we have this interlaced matrix of copy numbers, copy number states, and the binary change points. So it shows, for instance, for marker 1, you see that in cell 3, between the first bin and the second bin, there's a change in copy number, and this is what we capture. And this reduced lossy transformation is what lets us gain scalability. Okay, so so this would be the second ingredient, is that we're assuming perfect phylogeny, so that basically if there is basically the genotype or the pattern of presence and absence of markers for a specific cell would be and would have markers present that are between the shortest path from that cell to the roots. Okay, that's one way to think about this. And so this is basically, you see that there is a tree, and this is the corresponding latent marker matrix. And here's the point: if we impose a perfect phylogeny on the observed data, then again, this is going to be very brutal, and the inference becomes very difficult, and the robustness that we would want from a cancer phylogenetic method would be lost. But the point here is that instead of assuming the perfect phylogeny on the observations themselves, we assume it on the latent marker matrix. And you can think of it as a dual of the input that is presented, the binary input that's presented to the inference algorithm. And this allows these violations of the perfect phylogeny, but you still can infer your phylogenetic tree. Okay, next slide, please. Thank you. Okay, so I'll skip on the third part, which is the the MCMC move, the tree acceleration move that allows this fast inference algorithm, but it's on our bioRxiv. If you're interested, please, we can also talk about that a little bit more at the end. But here, this is the results that we have on the publication that we've submitted for sitka. And we're looking at 3 different data sets, 2 PDXs and a cell line. The main point here, again, is that on the left, you have the inferred tree, and on the right, you see this pattern of presence and absence sorted by the tree. And the block here structure is very well visible. Okay, and we have tests that show even on real data sets, which is often very difficult to verify, that we have a higher performance than existing methods. Okay, so this is sitka. And so once we have the phylogenetic tree, next move is to define clones. And very roughly, you can imagine that this is just the major clades in the phylogenetic tree. And we use, in this project, we've used copy number aberrations as a lineage, markers. So we're tracking the lineage of these cells. And from there, we can define clones that are shared across different conditions. So this is another thing that you get from a phylogenetic analysis. Now, from the experimental design and the phylogenetic analysis, you get a time series data. So you have the clonal prevalence of clones over time. Okay, thank you. So to model such a data set, we went back to population genetics, and we're using the Wright-Fisher model. So this picture on the right shows the Wright-Fisher model in a nutshell. Basically, you start with a number of types. So this is a clone level inference method. So you have clones at the beginning, and then you assume that the number of your clones are fixed at each time point. And at every generation, we play this process where we sample from the previous generation. And by this process of sampling, some of the clones in the previous one would be lost. And you would assume, and then you continue this process step by step. And you would assume the ones, so here the circle is the clones that have a higher fitness are more likely to be propagated forward in time. And the ones with lower fitness are more likely to be lost. Okay. Can we go to the next one? Thank you. So so that's the process. The computational method that we think of, or a statistical framework that we model this through, is the state space models. So you can think of that where observations, these are the shaded variables here in this probabilistic graphical model. So it would be y1 through to y sub t. These would be the observed clonal prevalences. So depending on what you're working with, here we're using normal distribution. So we're normalizing the counts, but you can use a multinomial distribution, for instance, or additional multinomial, and use counts. And there's z variables here, the z variables here are showing the hidden state. So we are allowing for some measure of noise. And what we track over time, again, is the prevalence of each clone. So Z1 is a vector valued random variable. And then the dynamics of this process is state space model is governed by the Wright-Fisher model, specifically of a diffusion approximation of it. And here is a version for, when you have basically 2 clones, and this is in the stochastic differential model syntax. And what you see there is basically there's the prevalence of a clone has 2 parts. There is a deterministic part, and if you say that S, that's the selection coefficient, that depends on the selection coefficient mainly, and there is this random walk behavior, which is in the second part. So we allow for some randomness in our model. Okay, we'll go to the next one. Thank you. So inference in this framework is is difficult. We have a transition density that is degenerate and intractable. So you don't have a closed form for simulating forward or simulating backward, or knowing exactly the likelihood for if you observe a state at the beginning and a state at the end. So we resort to simulation methods. And in a nutshell, the way that the inference works here is that you simultaneously simulate trajectories forward in time that are consistent with the observations. So this would be coming from experimental sampling, and also infer selection coefficients. And at the end, this is done in an MCMC fashion. So you get a posterior over your quantities of interest, specifically these selection coefficients. Okay, I think I might be time. Yes, all right.
Okay, so thanks, Sohrab. Thanks for briefing the two methods. So let me first tell you the story how we apply these methods on the biological system in real time on the single cell time series data. So first of all, I'll tell you the story from one of the triple negative breast cancer SS609 time series that was propagated till 10 passages. And from each time point, we collected single cell genomes for analysis. So after collecting more than 3,000 genomes from passage 1 to passage 10 and applying phylogenetic inference to the copy number genotypes, we were able to recognize different clonal groups that were present in this tumor. The heat map on the left side is showing the genotypes and the subpopulations where the vertical axis represents single cells and horizontal axis represent the genomic position. Now, having determined the genotypes, we can say what are the relative abundances of each clone by relationships by computing phylogenetic tree. Phylogenies show branch structure where shaded areas are representing clones. So we found that clones E, G, and H that were high, that had the highest high in abundance were closely related to each other and in the upper clade of the phylogenetic tree. So next we observed and look for the observed trajectories from first time point to the 10th time point. And we found that 6 clones, where clones E and H had the highest selection coefficient and exhibited growth from undetectable levels to around 59% to 32% by the 10th time point. And inferred trajectories showed that clones E and H had the high selection coefficients when quantified by fitClone, which Sohrab has just explained. And the reproducible dynamics of clones E and H were confirmed from the replicate transplants. So next we wanted to validate and confirm these findings, like how firm these measurements are by taking the idea, if we just retake the populations and reshuffle their prevalences and we run the experiment, are we going to see the same clones with high fitness? So, so I took early and late time point samples, remixed them in 2 different proportions and transplanted a new group of mice to rerun the time series experiments. And in one of the mixture B, we created bias by adding around 1/4 of the sample, like very small of that sample, which contains clone with high selection coefficient. We knew that the clones were in high abundance in the later time point. So in mixture 2, they were in around 1/4 of the quantity as compared to mixture A, where we had approximately 50% of each population. And interestingly, we found the same clones E and H showing high selection coefficients in both independent mixture branches, as were seen in the original time series at first place. Comparing the phylogenies showed the same results. That closely related clones in the upper clade, they were very closely related. And this like computed the phylogenies and see if you see in all the three. The first two are the mixture branches, and last I put again to remind you what the original branch was having the structure. So I still remember the day when we had that result, and me and Sohrab was so excited to see these consistent results. Yeah, so after having these in untreated setting, next we were curious to understand how this fitness landscape, with some clones having a fitness advantage in an unperturbed environment, how this fitness landscape will shape in the presence of chemotherapy. So I took 3 patients' tumor and propagated the Timeseries PDX in the same experimental design that I explained in the beginning, where after treatment, residual tumor was retransplanted and re-exposed to the drugs in a time series manner, along with drug holiday samples that were kept in treatment off state. And one of this tumor patient in PDX is triple negative SS609 that I was explaining in the I was telling the story. Without the drug perturbation, we propagated many replicates, and that was an extra untreated branch and many treated branches, and we acquired many replicates just to confirm what results we are having and are they consistent or not. And luckily we found they were consistent throughout the analysis. And the right panel shows tumor growth record, where later time points tumors were not responsive to the drug, and along with the upper panel showing the drug exposure in each cycle. So if you can see in the beginning first exposure, the tumor size was shrinking very rapidly, but in the later cycles, tumors started showing resistant behavior. So these are the results from triple negative SS609, and where upper panel showing phylogenetic trees at each time point, and the lower panel showing trees from drug holiday samples. So what we found that under continuous drug pressure, clone A from lower half of the phylogenetic tree started emerging and took fitness advantage under the drug, while the clone that was fit in untreated setting was eradicated. If you remember, clone E and H were present in the upper clade of the phylogenetic tree in the absence of the drug. So, so when we look at the drug holiday sample, they showed that indicated that initially when drug pressure was taken off at one time point, there was a reversal of clonal fitness, and clone A, it decreased in prevalence. But later, once the fitness landscape was fixed, there was no reversal, and even in the absence of drug, clone A was present, showing that platinum has a fitness cost. Next, we compared the features of resistant and sensitive clones and found they were having different genomic features based on copy numbers, such as clone A had copy loss of tumor suppressor RB1 and gain of oncogene SQLE. And so we concluded that the clones that had higher fitness under no drugs were eliminated, and the lower fitness clones were the precursors of eventual drug-resistant clones. We also noticed that for all the 3 TNBCs, at least one clone showed a high degree of clonal fitness, indicative of positive selection and non-neutral clonal dynamics, linking in principle copy number defined genotypes to differential fitness. So yeah, with that, I will summarize the results that copy number clonal structures can be used to link genotypes to fitness that we validated with mixture sampling. Clonal platinum resistance has a fitness cost, meaning one genotype was less fit than the precursor clone in the absence of the drug. And we found an inversion of the fitness landscape in all the 3 time series PD axes. With that, I would like to thank my supervisor, Dr. Sam Aparicio, and Dr. Sohrab Salehi. He was also my committee member and a collaborator, and for sure, Dr. Sohrab Salehi. So me and Sohrab, this whole project, like it cost a PhD for both of us. And at the end, when we were able to publish this paper, we both defended our thesis with success. And also, this was not possible without the single cell platform senior technicians from Aparicio lab. I only gave my single cell suspensions of the tumors to them, and they managed to spot them through fancy robots in the single cell chip. And of course, all the members from Aparicio lab and Dr. Shah lab. I'm very thankful to all of them. And thanks for listening to me today.
Thank you so much, Farhia and Sohrab, for sharing your work with us today. And just as a reminder, next we're going to move to our Q&A section. So you can either raise your virtual hand and ask the question directly to our speakers, or you can type the question in the chat box. So I was wondering that today you present data on treatments on triple negative breast cancer. I was wondering, in the paper you also talk about HER2 positive models, which also have TP53 mutations. However, their clonal landscape is a little bit different. I don't know what other factors are driving, what other factors besides TP53 mutations can explain that?
Yeah, that's a very good question. So H3 cell line, so in the beginning, when we were trying to see whether these models are, how efficient these models are working, so we had the Timeseries cell lines, and then I did some experiments by creating the mixtures of these cell lines from initial and late passages. So that cell line was a wild type, and then at the same time, we developed and created TP53 null cell line, and we propagated the same way. And then we did mixture of the fittest P53 null with the wild type. And we found in the consistent sampling and mixture experiments, the results that the clone that was having fitness advantage was taking over in the late setting. And that gave us the courage to go in vivo and actually look at the patient sample in PDX models. Back to your question for like what other factors. So all the 4 PDXs we used, they were also on the background of TP53 null, like they were having mutation in the tumor suppressor gene. So it could be that background that could lead to, that could be the additive factor for the fitness, but that I mentioned, like when we looked at the resistant and sensitive clone, they were having their own distinct features. So in the paper, basically what we are trying to say that to identify a clonal genotype based on copy number profile is underrepresentative before. So it could be some additional factors that are carried out along with this genotype. That could be epigenomic, that could be other transcriptomic state or SNVs that is participating and causing that clone for its fitness advantage.
Thank you. We have Christoph wants to ask a question. Christoph, you can go ahead and unmute yourself.
Hey, very cool paper and presentation. So my first question, I have 2 questions, but the first question is that you guys use the Fisher-Wright model with diffusion and selection. So I was wondering, one of the assumptions that I saw up there is that the effective population size is fixed. And since you're doing you could be doing multiple passaging. This is like, for instance, a bottleneck, which kind of breaks that assumptions. Do you think that you could address that?
Sohrab, do you want to answer?
Sure, yeah. This is an excellent question. And definitely, from a mechanistic point of view, assuming that the effective population size is fixed is, I think, is not correct. However, from a modeling perspective, we investigated this, so you can change this effective population size and have it in a large range, so an order of magnitude or two less and an order of magnitude or two more. And what you find out is that basically the ranking of the selection coefficients that you find are the same. And so the one advantage that we had there was that we had long enough and finely sampled enough time series to be able to estimate our selection coefficients. There is one issue there, and that's with the time scaling, and I think it shows it's a sort of basically a variable effective population size shows itself there, where you can when you get this. So when you simulate from this model, you might find out that it sweeps through the population faster than what you see in the actual model, or does it more slowly. So that's something that does happen. Yeah, I'm not sure how to address that.
Yeah, my other question was relating to the assumption of non-overlapping generations. Is that also kind of, I'm not too familiar with how like cancer cells grow, but would you think that that's a good assumption?
I would say that Farhia can speak best to the actual underlying biology, but I would imagine, and we actually do observe that wouldn't be the case. So it's still this is another modeling assumption, but I would say that one way of thinking about this is that this is a model that is that helps us do predictions, and it's doing a good job of doing these predictions. Yeah.
Thank you.
Of course.
There's another question in the chat. So how do you validate the direct involvement of CNV genotyping for cancer cell state transitions and drug effects?
Okay, yeah, thanks for this question. So again, back to what I answered in the beginning, that we are not claiming that this copy number variation is directly related to the fitness. So there could be definitely and possibly many other factors that are contributing to this copy number state. But the main thing, how we validated it with by creating another parallel time series branches, and by mixing different proportions of fit population along with unfit population, and then looking at many biological and technical replicates to confirm the findings. Because in the beginning, when we first saw these results, like this question arises in everybody's mind, like, is it the clonal genotype? Is it the copy number variation, which is the real cause? So yes, we could, we were able to validate it in many different settings, but again, we cannot claim that this is the only reason that is causing the ultimate fitness or selection for that population.